coding for things that have amino acids at the end of them. But it's probably just like what you get with poly A tails. What's one of the reasons you have a poly A tail? Protection. Protecting the end of the genome from the nasty exonucleases, which would just love to chew up the ends of your genomes, and that's what it does when it's degrading messenger RNAs in general inside the cell. So that's what this structure seems to be involved in, is sometimes adding to the genome, but mostly getting protection for the rest of the genome. What are the proteins which are made here? The important ones are, again, not surprisingly, a capping enzyme. Here, it's RNA1, methyltransferase, and then all of these other RNA viruses also have what? RNA-dependent RNA polymerases. So you have these. What else do you need? You remember back to way back when, MS2, Q beta. Got to make your virion, right? What else do you need to encode? You need to encode a coat protein. So coat protein is this RNA4 here. So that's all you really need. Something to cap, something to replicate, and a coat protein. So there are other proteins which are encoded here. The main thing has to do with movement, getting from one cell to the next. And so there are some what are called classic movement proteins. This RNA3 basically encodes one of those, and this other RNA4 does it as well. So movement proteins, capsid proteins, RNA polymerase, and capping. Where does this, sorry. In this case, well, 1A is RNA1, 2 is 2A. I can't remember that it's 2B, that's an RNA4A. And particularly in this case, I hate this nomenclature, and it's 1A, 2A, 2B. Okay, it just happens to be the structures that you have there. I would know what the activities are, but not necessarily the numbers. Again, I'm not going to know this till I write the exam, which will be late Tuesday night. As usual, right. So it'll be good timing. So where does replication take place? There are the regular viral proteins. Again, are RNA polymerase and proteins that are involved here in capping. These we're going to bind to your RNA. Again, it's got this 3' tRNA-like structure. One of the things that seems to happen with most of these single-stranded RNA viruses in plants is that they hide themselves to replicate. Why are they hiding themselves to replicate? Probably because cells, and particularly plant cells, are really good at getting rid of double-stranded RNA. In fact, RNA interference was originally discovered in plants, where you had double-stranded RNA, which would then suppress expression of different genes. So probably the reason that you have these special structures which get made during replication is probably just to protect this double-stranded RNA intermediate from getting degraded. There are another couple of things that you have to have in order to get normal replication here. Lots of translation proteins. This should sound really familiar and similar to which RNA virus that we've talked about so far? Not that I'm specifically testing on this midterm. Give you a bit of an idea. The one that I had just mentioned in terms of what minimal numbers of proteins you need. MS2, so the RNA bacteriophage. They also need translation proteins, cellular translation proteins, in order to get good replication and packaging to take place. So, number of these cellular proteins, again, particularly the translation proteins. EF3 is one of the initiation factors. And then there's a DNA J-like protein which is required. This is a protein folding enzyme, probably for assembly of the individual virus particles. So those are, again, not terribly different. What is different as far as these plant viruses is, again, how they get around. So movement protein, in the case of cucumber mosaic virus, is 3A. Other plant viruses also have movement proteins. And the best studied of these movement proteins is actually the TMV movement protein. But anytime you go to a virology meeting and people are talking about plant viruses, they're all about movement protein this, movement protein that, movement protein this. Because it's actually kind of hard to get around plant cells. Again, because they're pretty tough and they can't run away. So they want to be very careful about controlling the transmission of different things between different cells. So the plasmodesmata are the classic case of connection from cell to cell, because the plant does need to communicate between different cells. These plasmodesmata are really small and usually only allow very small things through them. These movement proteins, at least some of them, cause an expansion of the plasmodesmata so that RNAs, and particularly RNAs together with RNA-dependent RNA polymerases, can get through them. Apparently, full virions can't get through them. But this is your local transmission, plasmodesmata. You may remember back here, you read the table carefully, cell-to-cell movement. That's what's happening with 3A. Long-distance movement here is getting from these cells, which are the ones which are getting chewed up by the aphid, through to the rest of the plant. And that's the whole plant vascular system, xylem and phloem here. Most of you probably know way more about this than I do. But these are the cells that virus or the virus RNA needs to get into to be transported through the rest of the cells. So there are really two kinds of movement: cell-to-cell movement and then long-distance movement that have to be taking place here. There's one other thing which I forgot to mention about these proteins. Because you've got such a strong double-stranded RNA response, usually in plants, once they get infected by these plant viruses, there's also, not surprisingly, a plant virus protein, which is combating this. So again, if we go back to our table, this 2B protein suppresses RNA silencing. And this has been one of the big areas of research in a lot of these plant viruses, because RNA interference is a really big deal. Got a Nobel Prize a couple of years ago. Double-stranded RNA leading to silencing, cleavage of different RNA products. And so how plant viruses are dealing with this, how they've been an antagonist to this process, has been a really active area of research, and in fact a lot of it down at Oregon State. They were the first people to in fact discover this particular effect that was going on there. So that's our cucumber mosaic virus, all the chapter in about half an hour. Again, very, very wide host range in terms of what it can infect. Lots of different kinds of plants. The structure, nice, easy, boring, beautiful. Entry and transmission, it's all about making damage to the cell wall of the plant. Once it gets in, the movement proteins help it move to the plasmodesmata, but also get into the plant vascular system for moving things around. Any of you work on non-vascular plants? I think it's a really fascinating area for looking for viruses. Anybody looking for a great research project? Viruses of non-vascular plants. Yeah, in the back.
I just wonder, where's that vesicle, that budding happening that you showed?
Oh, the budding which is happening when you're getting replication, that's inside the cells. So, and it seems to be associated with the endoplasmic reticulum. There's, kind of a dirty secret of talking about this, is a lot of this work was done in yeast, which is not usually the host for these particular viruses. But as true with most positive strand RNAs, once you put it in, it will still replicate and translate. So if you can get all three of those RNAs into a plant cell, and you don't need the coat protein because it's not going to be spreading. But if you're looking at replication, and of course there are wonderful genetic tools, great cell biology tools for looking at this in yeast. It's probably happening in plants too. It's just a lot harder to study those individual plant cells. So probably the endoplasmic reticulum in the plants, in an individual cell. Yeah?
So when the plant's making the virus, the virus just goes somewhere and hangs out until the plant gets eaten by something.
So yeah, maybe I'll paraphrase your question, is how do viruses get spread from plant to plant?
Right.
Yeah.
So it just like builds up the quantity of virus and then just like waits to be eaten.
Right, waits, yeah, so again, waiting. This is again a little bit of an over-anthropomorphization here. But the real question is, again, how do you get transfer? How do you get transfer from one plant to the next? An uninfected plant gets infected from an infected plant. And it does basically seem to be, as you're saying, waiting. Waiting for some insect to feed on that plant and then transfer it to a different plant. So that seems to be the mechanism of getting transport taking place there. There are some examples of the virions being infective through roots. That's a kind of a controversial issue here. But it seems to be most cases you get very high amounts of virus production in these plants, humongous numbers, because that's what you have to have. You know, yeah, sure, the aphid's pretty big, but it's not going to be eating that much of the plant. And so you've got to have very high titers wherever that aphid happens to be eating in order to get transfer to take place.
And where does the virus build up?
It seems to build up in most cells. So many of the cells, particularly in leaves, is actively growing. And we'll get back to that when we talk about tobacco mosaic virus a little bit later on. But actively growing parts of the plant. So the leaves, the stuff that look yummy.
Where inside the cell does it like store or whatever?
Oh, so where do you get an accumulation of virions? Basic, so if you go back, sorry to go back here real quick, but your plant cells, most of them are filled with these vacuoles. And the cytoplasm is generally relatively small for a lot of these plant cells. If you look at a virus-infected plant, most of the cytoplasm will have virions in it. So it's pretty much in the virions, so just basically right underneath the cell wall. So as soon as that aphid comes in, takes a little snack, then it's got virions associated with it. But the numbers have to be really high to be able to do this and actually get proper transfer, because you need 3 of the virions in order to get this active infection. So it's really, again, it still blows me away that this has been evolutionarily so successful. It works. Okay, so I want to talk about a few other things here, some of the other viruses as well. One of the most fascinating aspects of a lot of cucumber mosaic virus infections is that many of them have these satellite RNAs which are associated with them. This is the whole sequence of some of those satellite RNAs. So how the heck could something like this actually have some kind of function in terms of disease, what's going on there? These were actually discovered way before people got really excited about small RNAs being regulatory and interacting with all kinds of other things inside cells. The plant virologists had known about these for decades. Oh yeah, sure, small RNAs are really important. They cause differences in terms of disease. How they cause differences, nobody really knows. But these short RNAs are really quite common. You've got some short RNAs that seem to cause a lower amount of disease, some which seem to exacerbate disease. Quite how that's happening. These RNAs are presumably also picked up through the capsids, so they just get encapsulated. They're replicated by